Autism spectrum disorder and the study of genetic chromosomal abnormality. A higher risk of ASD has been associated with duplications or deletions in certain areas of chromosome regions, notably 15q11-13 and 16p11.2. ASD is caused by a variety of circumstances, but evidence indicates both X and Y chromosomes may be involved. Abnormal amounts of chromosomes on sex chromosomes have also been connected to neurodevelopment disorders, including ASD. 6p11.2 deletion frequency. Research indicates that approximately 1% of autistic children have deletions or duplications in the 16p area. Rather than being inherited from a parent, this deletion or duplication frequently happens on its own around the time of conception. Although complicated genetic factors are frequently thought to be the source of autism spectrum disorder, in some cases with rates ranging from 2 to 4 involving specific chromosomal abnormalities such as those of the 15q, 16p, and sex chromosomes. How chromosomal abnormalities contribute to ASD. Gene dosage imbalance. Small DNA deletions or duplications known as microdeletions or microduplications cause an imbalance in gene dosage, impairing normal brain development and function and potentially contributing to the characteristics of autism. ASD syndromic forms. ASD-like symptoms and other health issues might result from chromosomal defects, such as those that impact particular chromosome regions. Examples include Smith-Magenis syndrome, Fragile X syndrome, Rett syndrome, Cornelia de Lange syndrome, Angelman syndrome, and 22q deletion syndrome. 22q11.2 ASD symptoms may be present in 22q11 deletion syndrome, which is linked to the deletion of this area. X chromosome. ASD is also linked to mutations or anomalies in genes on the X chromosome, including those involved in synaptic function. This is a karyotype testing chart linking chromosomes and DNA traits to karyotype testing. Finding chromosomal abnormalities in autism spectrum disorder. Karyotyping, although it may overlook subtler alterations, traditional chromosomal analysis or karyotyping can assist in identifying more significant chromosomal abnormalities such as deletions, duplications, or translocations. Microarray analysis. Chromosomal microarray analysis can identify structural differences in DNA, such as minor deletions or duplications that karyotyping could overlook. Fragile X testing. Patients with ASD are subjected to Fragile X testing. Here's a short video about genetic testing.
Today's discovery is brought to you by the VCU Medical Center. Every day, a new discovery. I'm Dr. Ray Lewandowski, and today's discovery is autism spectrum disorder testing. Patients with autism have abnormalities relating to their environment. They have speech abnormalities and repetitive movements. Genetics plays a very important role in most cases of autism. Most patients that are referred for autism spectrum disorder are children. We would interview them, get a very detailed history of what was going on with them medically. If we felt that they needed testing, the most common process is to obtain blood. Blood work that's done helps us determine if there's an identifiable genetic abnormality. The diagnosis of autism is typically made by age 3. One hopeful thing for parents of autistic children is the use sequencing, which hopefully will allow us to identify specific changes in genes that potentially could lead to therapy for autistic children. For more information,
Here's another short video about chromosome analysis and genome testing.
Parul Jayakar, Chief of Division of Genetics and Metabolism and Director Chief of Division of Genetics and Metabolism and Director of the Neurogenetics Metabolic Program at Nicholas Children's Hospital. Though we know enough about autism, we don't know everything, and we generally start with the common causes of autism. We do the chromosomes, we do metabolic workup. Then if we don't find anything and we strongly feel that yes, the child is not improving, and then we do something called the whole exome sequencing, where we sequence the known coding regions to try and obtain a diagnosis. We are also a part of a consortium through Sanford for the whole genome project, where we are now enrolling patients with the genome, and it's part of the Personalized Medicine Initiative program, where we analyze the genome on these undiagnosed, complicated cases, be it autism, seizures, or anything.
Genetic testing and raising awareness. Chromosomal abnormalities in people with ASD can be found with the aid of genetic testing, such as array-based comparative genomic hybridization, which detects copy number changes, and karyotyping, which examines chromosomes. Additional things to think about. Given the complexity of autism and its high level of genetic variation, many genes and genetic processes may have a role in the disorder in different people. ASD can also arise consequently environmental circumstances and other non-genetic reasons. Research on the interplay between environmental and genetic factors is ongoing. Raising awareness. Promote understanding and acceptance. Focus on the diversity of the autism spectrum and the unique strengths and challenges of individuals with ASD. Challenge misconceptions and stereotypes. Educate the public about autism, dispelling myths and promoting positive attitudes. Encouraging early intervention, early detection. Urge patients and medical professionals to keep an eye out for developmental delays and to get a professional evaluation if they have any concerns. Monitoring development. Monitor children's developmental milestones on a regular basis and get help if they are falling behind in any area. Availability of evidence-based treatments. Make sure the families and children with ASD have access to supportive resources and therapies that work, such as applied behavior analysis, ABA. Cooperation. Encourage cooperation between families, teachers, and medical professionals and other experts to guarantee that kids with ASD get all the help they need. Encourage young people with autism as they grow up. As they grow into adulthood, assist young people with autism by giving them the tools and encouragement they require to live freely and accomplish their objectives.